immune system. In this lecture, we're going to discuss the importance of the oral immune system in relation to dental caries. This system is of importance in the prevention of microbial oral colonization and in the prevention of penetration of any noxious substance through oral mucosa. The oral immune system can be divided to two types. The first one is called the non-specific oral immune system, and the second is called the specific oral immune system. Non-specific immune system is considered to be a helping factor for the specific immune system. The specific immune system is by the action of immunoglobulin, while the non-specific immune system is by the action of different enzymes and glycoprotein, as I'm going to discuss in the following slides. The non-specific immune system lack any immunological memory and do not subject it to any specific stimulation in comparison to specific immune system. The first non-specific immune factor is the lysozyme enzyme. This hydrolytic enzyme arises from both major and minor salivary gland, gingival clavicular fluid, and salivary leukocytes, and also it is secreted in addition to saliva. It is secreted in all body secretions like tears, nasal secretion, etc. It acts by lysis of gram-positive and gram-negative bacteria as it hydrolyzes specific bonds in the bacterial cell walls. It also attacks bacteria through aggregation of cell suspension of some of bacterial species. An important action of this lysozyme enzyme is the enhancement of the action of immunoglobulin A. So this enzyme is considered to be a link between specific and non-specific immune system in the oral cavity. The second non-specific immune factor is sialoperoxidase system. In this system, we have peroxidase enzyme. This enzyme can catalyze some of bacterial products, which is known as hydrogen peroxide, with thiocyanate. The end result of this reaction is formation of hypothiocyanate. The hypothiocyanate is toxic to bacteria. The hydrogen peroxide, that is bacterial products, is considered to be toxic to human host protein and cells. So actually, peroxidase enzyme or this system can protect the human host protein and cell from the toxicity of hydrogen peroxide. Sialoperoxidase system can interfere with one of bacterial metabolism, which is known as glycolysis. By this, this enzyme can control bacterial and bacterial metabolic products. Lactoferrin is the other non-specific immune factor in the oral cavity. Lactoferrin is a glycoprotein with 10% of carbohydrate. It acts by chelation of iron, the essential nutrients for bacteria, and also may have direct bactericidal effect. Mucins is another oral immune factor, and it affects the viscosity of saliva. Some studies reported that with increased viscosity of saliva, there is increase in the agglutination of bacteria and prevention of its adhesion to oral mucosa. By this method, we can reduce the severity of dental caries. But with increased viscosity of saliva, there is a reduction in the clearance of food and carbohydrate from the oral cavity. Therefore, some studies reported the opposite. It means with increase in viscosity of saliva, there is an increase in dental caries. So still, the effect of mucins in relation to dental caries is in need of further studies. Proteins in the saliva, like staphyline, proline-rich protein, and histidines, have some relation to dental caries. These proteins originated from saliva, but they are going to be present in the acquired pellicle. In the intake of carbohydrate, there is a reduction in the pH of saliva and release of ions from the outer enamel surface, like calcium and phosphorus. Sometimes these ions, when present in the oral cavity nearby teeth, they're going to be crystallized in form of dental calculus. These proteins going to prevent recrystallization of these ions in form of calculus and enhance its remineralization to the outer enamel surface. So we can say that staphyline, proline-rich protein, and histidines, the main action is by enhancing remineralization by prevention of recrystallization of free calcium and phosphorus ion in form of dental calculus. Specific immune system. This system acts by the action of different types of immunoglobulins like immunoglobulin A, G, M, D, and E. In normal condition, when there is no inflammation in the oral cavity, the only immunoglobulin found in saliva is the secretory immunoglobulin A. In presence of gingival inflammation and through the gingival exudates, serum immunoglobulin is going to be found in the oral cavity in addition with secretory immunoglobulin A, like serum immunoglobulin A, serum immunoglobulin G, M. In normal condition, the type of antibody or immunoglobulins present in oral cavity is the secretory immunoglobulin A. The difference between serum and secretory immunoglobulin A is the presence of additional glycoprotein known as secretory component. The immunoglobulin is synthesized by immunocyte cells, that's plasma cells and mast cells, while the secretory component originated in the glandular secretory epithelium. Secretory immunoglobulin A is found in all body secretions. That's in addition to saliva, it is also secreted in tears, bronchial secretion, and milk. Immunoglobulin G is the predominant immunoglobulin in blood, lymph, peritoneal fluid, and cerebrospinal fluid. It is the only class of immunoglobulin that can cross the placenta and is responsible for protection of the newborn for the first months of his life. The general function of immunoglobulin in relation to dental caries involves first, elimination of bacteria from the oral cavity. Second, interference with the cariogenic potential of cariogenic bacteria. This is through inhibition of bacterial adherence, by blocking of adherence determinants, or by agglutination of bacteria. Second, through inhibition of bacterial enzyme systems necessary for metabolisms and growth of these bacteria. Immunoglobulin A has anti-inflammatory action in mucosal tissue, while immunoglobulin G has the ability of bacteriolysis and also has the ability of induction of inflammation through chemotaxis and opsonization of these bacteria, or through phagocytosis by the polymorph nuclear leukocyte. It's worth to mention that immunoglobulins may transfer from the mother to the infants by breastfeeding. So always the mother are encouraged to breastfeed her child, especially in the first months of his life, as the infant may receive about 1 gram per day of secretory immunoglobulin A, and this is very important to interfere with the establishment of mutant streptococci, the cariogenic bacteria, in the earliest part of the child life. The oral cavity is under the influence of the oral immune system, involving first humoral immunity by the action of salivary and serum immunoglobulins, and by the action of cellular immunity through the polymorph nuclear leukocytes that enter the oral cavity via gingival exudate. However, the indigenous flora can survive this immune system. This gives an idea for using a vaccine against cariogenic bacteria without affecting the indigenous microflora. Immunization is a process involving the exposure of the host to an antigen. Therefore, stimulate antibodies of immunological memories. The idea of preparing antigens to stimulate oral antibodies was back to 1930s, and the antigen used was lactobacilli. Then in the 60s of the last century, the mutant streptococci become the target in the experiment, and they prepare antigen from this bacteria for stimulation of oral antibodies. Vaccination, therefore, is a preventive measures that a susceptible host is artificially prepared to resist infection. The antigen used in vaccination should provide a maximum preventive activity with minimum undesirable side effect. So in preparing a vaccine, against dental caries, the host is exposed to an antigen taken from cariogenic bacteria. It will lead for stimulation of antibodies. Thus, reduced dental caries by elimination of cariogenic bacteria. Immunization against dental caries is still experimental and conducted on animals only. For this experiment, we need first to prepare the antigen, either using the whole bacteria or using certain bacterial enzymes or certain cell wall proteins. The induction of this antigen to the animal can stimulate either secretory immunoglobulin A or serum immunoglobulin A. There are two methods of stimulation of secretory immunoglobulin A. The first one is called the direct stimulation method, and this is by injection of the animal with antigen in the vicinity of parotid and submandibular gland. Again, this injection will lead to stimulation of secretory immunoglobulin A. Another method may lead to indirect stimulation of secretory immunoglobulin A by feeding the animal the antigen, and the antigen will come in contact with the gut-associated lymphoid tissue in the small intestine. By this method, secretory immunoglobulin A will be secreted in the saliva and all other body secretions like tears and milk. The other method of vaccination lead to stimulation of serum immunoglobulin, precisely immunoglobulin G, in addition to immunoglobulin A and immunoglobulin M. This is achieved by the subcutaneous injection of the animal by selected antigen. The immunoglobulins will reach the oral cavity through transduction with clavicular fluid. Although vaccination is considered to be a cheap method for protection against dental caries, there are still questions regarding their safety and effectiveness for a long period of time. The stimulation of serum immunoglobulins is considered to be effective and lasts for a long period of time. However, there is a possibility of cross-reaction with the heart muscles as reported by some experiment, and it has been found also that stimulation of serum immunoglobulin may increase the inflammatory reaction, thus may increase the penetrability for antigen to deeper mucous tissue, jeopardizing the gingival protection against plaque antigen. While the stimulation of secretory immunoglobulin A, although it is safe, however, it only lasts for a short period of time. Thank you for your kind attention.